Thank you very much. Thank you very much, Sushmita. It's a great pleasure to be here. Thanks for the very kind introduction. So I worked on the problem of population structure 24 years ago now with Jonathan Pritchard and Peter Donnelly. And if you pay attention, you'll see that I'm kind of still doing the same thing. I just want to assure you, I didn't spend all 24 years doing the same thing. This is like kind of circling back now, I feel like a little to some of these ideas and maybe trying to improve on them, because I feel like some of the stuff we did then certainly been useful, but I think we can do even better. So it's kind of my goal here is to tell you about that. So I'm talking about matrix factorization, and you may have an idea of what matrix factorization means, but it turns out I've learned that different people have different ways of thinking about this. So I want to tell you about the way I'm thinking about it to try and get us on the same page. So the basic idea behind matrix factorization methods is that given a data matrix X that's n by P, you factorize it into two smaller matrices, which I'm going to call L and F. So L is n by K and F is P by K, and think of K as being probably kind of smaller than N or P, although one of my goals is to make K a bit bigger than maybe is often traditionally used. So maybe K might be 30 or 50. Something like that, maybe even 100. But it's small compared with N and P. So, a simple way to try and find such matrices is to, say, try to minimize the sum of squared errors between the matrix X and this, quotes, approximation, LF transpose. And later we're going to be talking about different kinds of constraints on L and F, such as maybe non-negativity. And just to give some examples of methods that fit into this framework that you might have heard of, principal components analysis, or more precisely, truncated singular value decomposition is one method of solving this problem with a certain set of constraints on L and F. And non-negative matrix factorization is another way of solving this problem, specifically with L and F being non-negative. Okay, we'll talk a bit more about why you might want to make those assumptions later. So that's the basics of matrix factorization, but the way I want you to think about this is that every matrix factorization effectively decomposes your data X into a sum of K components. So it's simply an algebraic fact that you can write the matrix product LF transpose as the sum over K equals 1 to big K of these outer products of vectors. LK here is the kth column of L, and FK is the kth column of F. Right? So any matrix factorization is actually doing a decomposition of your data. And sometimes, if you're lucky, or maybe if you're careful, or if you use the right approach, these individual components, these capital K individual components, correspond to something that's interpretable physically or scientifically. And this, sometimes people have different ways of describing this idea that the parts are interpretable. I've sometimes called it interpretable matrix decomposition, but I found that people didn't resonate with people what that would mean. So I've switched to usually using this term parts-based representation, which was introduced by Lee and Sung. So what they mean by a parts-based representation is a matrix factorization method where each part corresponds to something you can kind of interpret. Okay. So my goal is to develop matrix factorization methods that produce part-based representations. That is, they produce a decomposition of your data into a small, capital K number of parts, each of which has some kind of scientific interpretation that tells you something ideally. Okay. So what affects whether matrix factorization methods produce parts-based representations or not? That is, what affects whether the parts, the decomposition, is interpretable? I think several things influence this, and I'm going to try to talk briefly at least about all four of these today. So one, and the major, the first approach that people use to try to induce parts-based representations perhaps is non-negativity. So Lee and Sung pointed out that non-negativity can make these decompositions more interpretable. Sparsity can also help. Orthogonality may influence anyway, whether it helps or hinders, might depend on exactly how it's used, but it definitely affects things. And the data transformation you use can also affect things. It's a great ringtone. So let's start with non-negativity. So this is the example that Lee and Sung gave. So I'm using their pictures from their paper here. They considered doing matrix factorization on images of faces. Okay, so in each face is a square grayscale image here. But that's not the matrix that's being factorized. The matrix that's being factorized, the rows are faces and the columns are pixels. So you take your image and you vectorize it. You just give it you turn it into a long string of pixels, and that becomes a row in your data matrix. So each row in your data matrix is the pixels from a facial image, vectorized. Okay, and then you factorize that matrix, and then you visualize, you take the rows of or columns of your L and F, and what they become is a representation of essentially each image in your database becomes represented as a non-negative combination of, these parts. And so this picture here shows what those parts are. And so what you can see is that each of these images is pretty sparse and it's non-negative, and it the argument is that each one corresponds to parts of a face. So maybe I can highlight this one down here, looks like two eyes, maybe with some eyebrows involved. And this part here is maybe two eyebrows. And this part is part of the chin here. Maybe this is part of the nose. So the idea is that each image in your database can be represented as a sum of these parts. You can represent a face as a sum of a part that captures the eyes, a part that captures the nose, a part that captures the ears. That's the idealized concept they're trying to get across. And you might say, well, does it really work? I think when you contrast it with what principal components analysis does, you can see that it's definitely doing something different and arguably more interpretable part space. So this is what PCA does. So PCA also represents each face as a sum, as a decomposition, a sum of things. But the difference is that now these sums of things can be positive or negative, right? They don't have to be positive. And the result of that, allowing that additional flexibility, is that things can cancel out. And that makes it more complicated to think about and less like building up things as a sum of non-negative parts. Okay, so the first principal component here in the top left is a kind of an average face. So the idea is that you represent a face by an average face plus these kind of other weird things which are a little hard to interpret but are modifying that average face in various complicated ways, right? And so that's the difference between the PCA representation and the non-negative matrix factorization representation. You can see arguably these parts are pretty hard to interpret apart from the first principal component. This is a general kind of empirical observation, let's say, that principal components analysis just usually does not produce parts-based representations. That is, if you look at the individual principal components, if you're lucky, the first one will be interpretable to you. If you're very lucky, the second one will be interpretable to you. But by the time you get to the third principal component, it's usually not capturing something that's kind of scientifically separately interpretable. It's usually capturing some combination of things, which is what makes it hard to interpret. Okay, so this was Liansung's argument that non-negative matrix factorization can produce more parts-based representations, more interpretable interpretations. And they showed examples on faces. In 2017, we became interested in applying these ideas to gene expression data. So I just want to show you that kind of more genomic-y example here of a parts-based representation. What do you get if you apply, we called it a grade of membership model, but it's actually a synonymous essentially to non-negative matrix factorization, or also it's essentially the same as what people call topic model or closely related latent Dirichlet allocation. So these are all basically the same thing, I'd say, with minor differences. So we applied it to these data on gene expression from the GTEx project. So the data that we're factorizing here are donors or samples, about 1,000 by genes. And these samples come from 50 different human tissues. So we've got 1,000. So our data matrix is 1,000 by 10,000, let's say, for the number of genes. And essentially, we're applying non-negative matrix factorization to the matrix, or a normalized version of it. So you can interpret xij over the xi plus here as the relative expression of gene J in sample I. And so L and F now become interpreted as, you can think of L as representing the membership of each sample I in each component K. And you can think of F as representing the relative gene expression of gene J in component K. So each component you can think of as being a set of expression levels at the 10,000 genes. So that's what F is representing, a bunch of expression levels. And you're trying to find those kind of canonical vectors of expression levels such that you can represent each sample as a combination of those. And then we visualize, one nice thing about non-negative matrix factorization is it often leads to nice visualizations. Non-negative things can make it easier to visualize. So here, what I've done is produce a stack bar chart, sometimes called a structure plot because it's widely used to visualize the results from a piece of software called Structure. But this is a structure plot or a stack bar chart where each column here is a sample. So there were 1,000 samples. So each column is a sample. They're organized by which tissue they came from. So the brain samples are over here. These are transformed fibroblasts here. There's blood samples here, so that each kind of, the samples are organized by where they came from in the human body, and then they're colored by their membership in the different components that we're identifying. That's this L here, the membership. So what you can see here is that most samples are assigned primarily to one component. That is, there's a majority color. Each one has a color that's kind of dominating, but there are also other colors. So if this were clustering samples, every sample would get one color. Right? It's not really quite clustering. It's assigning each sample partial membership in different, quotes, clusters or topics or whatever you want to call them. Okay, so that's the essence of this representation, is that each sample is being assigned partial membership in these different topics. So how can we try to interpret these topics or components? Well, one way we can do it is just look at which tissues they're falling in. So of course you can see, for example, this orange one here is mostly in the muscle skeletal tissues, and this light blue here, for example, is in the two skin tissues. There are two sun-exposed and sun-not-exposed skin. So we get some insight into what they're capturing just from that. But you can also look at the other part of the component, which is which genes are being highly expressed in these components, because each component also has a set of gene expression values associated with it. And so maybe, maybe I can just pick out one thing in this. You can actually spend a lot of time staring at this and looking at what's going on to see the finer scale structure. But let me just pick out one thing here, which is that the tibial artery tissues here have some partial membership in this pinkish. On my screen, it's salmon. We called it, I think, is salmon component. And that's also present in greater abundance in these esophageal tissues and related tissues. And if you look at the genes that are related to this component, they're genes that are, sorry, I was trying to remember what genes they are. They're genes that are related to the smooth muscle tissues. And so I didn't know this, but when you go looking, it turns out arteries in general, and particularly arteries associated with muscles, express smooth muscle genes because they help contract, they help regulate blood pressure, I think, and blood flow. So this kind of contraction thing that's going on is shared between these esophageal tissues and these artery tissues. So that's the kind of insight that you wouldn't get if you just clustered things, right? If you just clustered things, you would miss these shared components that are present in multiple tissues. And actually, I won't spend any longer on this, but there's several examples of that where you see it's capturing some kind of commonality between tissues that might or might not have been thought to be similar. I think we had about 30 components here. You could ask me, how did you choose the number of components? Actually, that's the most common question I get in presenting these kinds of things. So here we chose it based on how many colors we could manage on the thing. I think that's my favorite way to choose the number of components, is how many colors can you distinguish. Okay. So that first part is really trying to highlight, if you haven't used non-negative matrix factorization, it's trying to introduce you to the idea that this can produce more interpretable decompositions than, say, principal components analysis, and maybe also in some cases than simply clustering things. And I think it's a really good start. But there are other things one can do to maybe make these decompositions even more interpretable in some cases. And so now I'm going to talk to you about a method we developed that allows incorporation of sparsity into the LNF, and how that can help make things more interpretable, more parts-based representations. So I'm going to start by just showing you the example and then tell you how we did it, how the method works. So the example here, it also comes from the GTEx data, but it's slightly different. It's a different processed version of the data, where the GTEx data is used to identify what's the effect size of a bunch of expression QTLs. These are genetic variants that are associated with gene expression. So an eQTL is a genetic variant that's associated with gene expression. So each eQTL has an effect size, which denotes how much does gene expression change between the two alleles of that eQTL. So the data here are the effect sizes in a bunch of different tissues for each eQTL. So the rows are eQTLs, genetic variants that are associated with gene expression, and the columns are tissues, and the ijth element is the estimated effect size of that EQTL in that tissue. So the idea is, by making a matrix factorization, we're trying to understand how do the EQTL effect sizes in one tissue correlate or relate to EQTL effect sizes in other tissues. We're trying to understand that structure of the effect sizes. So you can think, again, here you can think of LIK as representing the quotes membership of EQTLI in component K, and I put membership in quotes here specifically because in this particular case, we're not going to assume L is non-negative. We could have, but we're not. Okay, so membership here can be kind of positive or negative. That's because EQTLs, depending on which allele you choose, could increase expression or decrease expression. So that's one way of thinking about it. And then F is the effect size in tissue J due to component K. So again, F can be positive or negative. But we're going to, instead of using non-negativity to try to get part-space representations, we're going to use sparsity to get part-space representations. So this is the results of running our sparse method, which I'll tell you more about in a moment. So let me just try to, each column here is a tissue. The colors you don't need to worry about too much, except I'll just note that the yellow here is a bunch of different brain tissues. So that's helpful for understanding things. So this first factor or component here, all the tissues are showing a positive effect, pretty similar in all tissues. So what this first factor's representing is the shared common effect that's common to all tissues. So if an EQTL was loaded or had membership only on that component, it would essentially be an EQTL that's shared across all the tissues at a pretty similar effect size. And then the next factor captures essentially the brain tissues versus the non-brain tissues. So that can capture the fact that there are quite a lot of EQTLs that have a rather different effect size in brain than they do in the non-brain tissues. This third factor is completely different. It's very sparse, and it's essentially loaded only on the blood tissue. That's this one over here. So this is capturing the fact that there are some EQTLs, not that many, but some, that have a much different effect in blood than they do anywhere else. Perhaps a much stronger effect in blood than anywhere else. And then this third factor, or fourth factor rather, here is interesting. I think it captures the two skin tissues. So these blue ones here are the two sun-exposed and sun-not-exposed tissues. And another tissue that's part of the esophagus. So presumably there's some kind of cell type sharing between these tissues that causes these three tissues to share EQTL effect sizes, that is, have more similar EQTL effect sizes, sometimes anyway, for some examples. And so these are more, you can see they're sparse, and they're capturing other more tissue-specific effects. If you go on down, there are dozens of factors that capture sometimes tissue-specific effects, sometimes maybe more compelling, more convincingly from the point of view of arguing the method's doing something sensible. It will capture a small number of related tissues, like two different heart tissues. And that's capturing the fact that those two heart tissues have more similar effect sizes than others, and there are some EQTLs that have much stronger effects in the heart than in other tissues. Just to contrast that, this is what you get if you do something closer to PCA. It's not quite PCA, it's this software soft impute, but essentially you can think of it as close to PCA and no sparsity assumption. And what you see is that the first component's very similar to our first component, so it's kind of interpretable. It's kind of that shared effect. The second component is somewhat similar. It's capturing the difference between brain and non-brain, although there's a bit more, it's maybe not quite such a clean story here. Like this is pretty much brain, non-brain, and with the testis and the pituitary kind of showing some commonality with brain, whereas this one's got a bit more going on. But where the real difference obviously comes is in the factors 3, 4, 5, 6, which really show kind of almost random patterns of change across different tissues. No, it doesn't pick out the two skin tissues or tissue-specific things. And this is kind of, as I said, this is kind of typical, I think, of PCA type methods, that you might interpret the first two components and then you give up. Okay, so I want to tell you now a bit about the method that we developed to do this kind of thing. It's a very flexible method. It can do more than just introduce sparsity. So this is just the matrix factorization model written. X is the sum of these k components plus E. And the empirical Bayes idea is that you put prior distributions on the components of L and the components of F. So there's a separate prior for each K, for each component. There's a prior distribution on the elements of L, and there's a different prior distribution on the elements of F, the corresponding elements of F. And both those prior distributions we would like to learn from the data. So think about these prior distributions as maybe capturing the sparsity or not of each component. And we're going to learn that sparsity as well as learning these L's and F's from the data. So we're simultaneously wanting to learn the priors, the G's, and the loadings and the factor values, L's and F's. And if the learned priors G are sparse, then that will induce sparsity. Or when I say sparsity here, I'm not really talking about exact zeros. I'm talking about the fact that many of the elements will be very close to zero. So it's kind of a soft version of sparsity I'm talking about here. So sparse priors will induce that kind of soft version of sparsity on the elements of L and F. Okay, so that's the goal. Okay, so to explain how we do it in a bit more detail, how we achieve this goal, I need to simplify a bit further. Okay, so this is a simpler version of the problem where you just have one L and one F. So I've set capital K to 1 just to get things started. So now I've got this problem. I want to represent X as LF transpose plus some error, where the L's come from some prior GL and the F's come from some prior GF, right? And the error terms are normally distributed with some column-specific variance in this case. Actually, that's another thing that I kind of sweeping under the rug. Different methods can assume whether columns have column-specific variances or whether there's a shared variance and blah, blah. That actually can affect results as well. So just a side note. But my goal is to tell you about how we estimate GL and GF and L and F in this model. Okay, so actually this is still hard. So I'm going to simplify I'm going to tell you about an easier problem that's related that I can solve, and then I'm going to go back and tell you how I use this method to solve the matrix factorization problem. So this easier problem is called the empirical Bayes normal means model. Okay, so what's the empirical Bayes? This will look like a different model for a moment, but you'll see how it's related in a moment. Okay, so the normal means model is where you have observations, which I'm going to call Y, of different means, theta, with some known variance S. So here the S's are known, but the thetas are unknown. So you've got a bunch of measurements that are noisy measurements of some underlying thetas, right? So you've got a whole bunch of, imagine you've done a differential expression study where each observation is a measure of the change in expression of gene I between two conditions, and theta I would represent the true change between the conditions, or say log-fold change, theta I, and yi is the observed log-fold change. So it's this kind of thing where you've got a bunch of noisy measurements of some underlying things that you'd like to estimate. Happens all the time in genomics, actually. So this model is kind of useful in and of itself. And the empirical Bayes part comes in because we're going to assume that the true values, the thetas, come from some prior distribution G that we're going to estimate from the data. So, okay. So this is a much simpler model. And it turns out that you can fit it in 2 steps for a whole bunch of different prior families, which I've called curly G here. So the details will depend on which prior family you choose, but there's a whole bunch of flexible prior families for which you can easily solve these two problems. That is, we can estimate G by combining data over all the observations to estimate the prior by maximum likelihood, essentially. And then, having estimated G, we compute the posterior distribution of the individual thetas given that estimated G. And that's where the essence of empirical Bayes is that we fix G to its maximum likelihood estimate, or a point estimate, G hat. So that's where the simplification comes in over maybe doing something more, quote, fully Bayes, right? Where you might try to take account of uncertainty in G. The empirical Bayes method simplifies things by ignoring uncertainty in G, just getting a point estimate for G, but then getting posterior distributions for theta. Okay. So the idea, again, is if the true thetas are very sparse, then hopefully the estimated G will reflect that and be very sparse. And so then this posterior distribution will reflect the sparsity of the estimated G, and so the estimated thetas will also be sparse. That's the essence of this one. Okay, so I'm not going to talk any more about the details, but you'll just have to take my word for it that this is actually quite an easy problem to solve. We have good software for solving this for a whole bunch of different families G. Okay, and if you're interested in the details, there's a software package, EBNM, and a paper on my website that's to appear in Journal of Statistical Software. Okay, so back to the matrix factorization problem. How does this help us? This is the matrix factorization problem. So the idea is essentially, just for a moment, imagine we knew F and we only wanted to estimate L. If you imagine we knew F for a moment, then it turns out that estimating L is essentially an empirical Bayes normal means problem. And just to kind of, one way of seeing that is to post multiply by F, so you get XF equals L, F transpose F plus EF, and F transpose F is just a number, right? So it's a scalar. And so what you get is, if you call this Y, you've got Y is L is, sorry, Y, the mean of Y here is L. Well, again, if you divide by F transpose F, sorry, you have to do that as well. Call that Y. So Y is XF divided by F transpose F, and it has mean L and some variance that you know. So essentially, the idea is that if you knew F, estimating L is just a set of n numbers, and they have a prior GL, and that becomes the normal means problem. So you can solve it by applying the empirical Bayes normal means software. And that might suggest to you, well, some kind of iterative scheme where we alternate between doing this for L, assuming we know F, and then doing this for F, assuming we know L, and going back and forth. And it turns out that if you get the details right, that corresponds to making a variational approximation to the posterior for L and F. So, and that connection establishes an objective function and guarantees convergence of this iterative approach. So the idea is that you can make some kind of rigorous justification of that intuitive iterative approach to the problem. And then that's for k equals 1. What you can do for k equals 2 is you kind of estimate it for k equals 1, and then you remove that estimate from your data and repeat. And this is a commonly used strategy in matrix factorization methods, sometimes called deflation, where you estimate one component, remove it, and repeat. And that's a greedy procedure, essentially. You can actually go back, if you don't like greedy procedures, you can actually go back once you've estimated k of them, you can go back to the first one and re-estimate, which can sometimes help. Actually, the greedy approach works pretty well in a lot of cases and gives you a nice quick answer. But if you have time and inclination, you can go back and repeat, and sometimes it does help refine some of the factors. Okay, so this is all in a paper with my student Wei Wang from 21 in the Journal of Machine Learning Research. So if you want the details, you can go and look Okay, so the kind of one of the nice things about this approach is that you don't only get to induce, say, sparsity, you can induce other things. Essentially, it's a very flexible approach that just requires you to be able to solve the empirical Bayes normal means model for a prior family that you want to use. Once you can solve that problem for that prior family, it just plugs into the matrix factorization iterative scheme. So if you say don't like any of the prior families we've got implemented already and want to implement your own fancy prior family, you just have to implement a solver for the normal means problem, and then I can assure you, a much easier problem than the matrix factorization method. Then you plug it in and the iterative approach does the rest for you. So it's kind of very flexible. When you use sparse priors or potentially sparse prior families, you're learning the level of sparsity automatically from the data as part of the fitting procedure. So you might want to contrast that with an alternative approach to inducing sparsity, which is to put penalties on things. Okay, so if you put a penalty on things, remember we have a different prior for each L and each F, and that's potentially desirable because some components may be sparser than others, and L may be sparser than F, or F may be sparser than L. We don't necessarily know in advance. So if we have K components, we have 2K sparsity inducing priors. So if you want to put penalties on these things, you end up with 2K sparsity inducing penalties. And tuning those by cross-validation becomes tedious. Right? Tuning one parameter by cross-validation is not too bad, but even two is kind of a pain because of the need to do a grid of pairs. So I feel like this matrix factorization setting where you really do want these different sparsity, potentially different sparsity levels for different factors, is a place where the empirical Bayes approach, which kind of automatically tunes things by solving an optimization problem rather than by cross-validation, has an advantage over penalty-based approaches. It's a variational approximation, and so it typically tends to be quicker than Markov chain Monte Carlo. You can solve pretty large problems easily. And I'll just mention that if your matrix is sparse, which it often is in single cell, say, genomics applications, then the computation scales only with the number of non-zero entries in the matrix. So that's nice. And my former student, Jason Wilverscheid, has implemented some of these ideas, especially the sparsity, the more efficient computations in a software package, Flashier, which is on GitHub, and although the paper is in preparation, the software has been around for a while and it's ready to use if you want to use it. Okay, take a break from methods now, back to some of the ideas. We've looked at non-negative matrix factorization, we've looked at sparsity. A natural desire might be to try and combine those two ideas. Let's have non-negative and sparse. And now using these empirical Bayes matrix factorization methods, it's easy to do that. We just use a prior family that's both non-negative and sparse. And the specific one, the simplest one that we use is this mixture of a point mass at zero and an exponential, right? So the exponential's non-negative and the point mass at zero induces potential sparsity. So it's a prior family that's a mixture of, sometimes called a spike and slab, where the spike is at zero and the slab here is an exponential. So prior distributions in this family are non-negative, potentially sparse, and so this provides us with a version of matrix factorization that's non-negative and sparse, and we call it empirical Bayes non-negative matrix factorization. So let's talk a little bit go back to the single cell RNA-seq application that I briefly mentioned. I think that non-negative matrix factorization based approaches are obvious candidates for providing interpretable parts-based decompositions of single-cell RNA-seq data. So the data that you observe there are counts. Why? So that the rows in my case are cells, the columns are genes. So Yij is how many times did I see gene J in cell I? Okay. And I just want to mention some kind of, I'm going on to a third part here, which is transformations of the data. So the topic model stroke non-negative matrix factorization ideas that I showed at the start of the talk, they're traditionally usually applied directly to the counts. So they model the counts as essentially Poisson or multinomial. However, an alternative approach is to transform the data. So this is a commonly used transformation in single cell data. So it's a shifted log transformation. This is a normalized version of the count. And then because this normalized version can be 0, you can't take a log of it, but you want to take a log of it. So what do? Well, you add something to it before taking the log of it. So this is a shifted log transformation that's commonly used in single cell data. And so an alternative to doing the topic modeling, which models the counts directly, is to do a transformation and then use a different, say a Frobenius norm NMF or empirical Bayes matrix factorization on these normalized, on these transform counts. Okay, so I'll just mention that while statistically it feels much cleaner to directly model the counts, something more subtle is going on here as well, because it depends what you're assuming is additive, right? In the topic model, it's the counts that, the mean of the counts that you're assuming is adding up. So you're decomposing that mean, but it might actually make more sense to decompose, to assume that components act multiplicatively. That is, if something is present in a given cell, maybe it doubles the gene expression of certain genes. Some factor, some biological factor comes on. Maybe it affects gene expression in a multiplicative way. In fact, that's usually how we model the way that factors that we measure act on gene expression through log fold changes. So fold change is the multiplicative side. So from that perspective, you might prefer to think on a log scale, and this is closer to the log scale than the topic model. So this is maybe a subtlety, but when you see people applying non-negative matrix factorization to single cell data, they might be doing something that's directly modeling the counts, or they might be doing something that takes a transformation, and it turns out that affects the results quite a lot. So that's another part. That was the third part. We've had non-negativity, sparsity, transformations also affect whether you get a parts-based representation. Okay, so just to illustrate, at least, sometimes it can be hard to say method A is better than method B, but at least we can show that method A is different from method B. So I'm going to focus on that. I am going to kind of argue that our method's better, but the most important argument is that these things are different, so you should pay attention to, maybe you should do all three and see which one you like best or gives you the most interpretable parts. That's fine by me, because I don't think of any of these as right and others as wrong. I think of them as giving you different representations of the data. Okay, so these data are from a paper by Montaro et al. They're mouse epithelial cells. And I just want to, the only thing I'm going to highlight really here is that the kind of main focus of the paper and contribution maybe is to discover a new cell type that they name pulmonary ionocytes. So that's just going to come up in a moment, because our method is going to give you a better view of those. It's going to pick those out a bit more cleanly than the other methods. So this is, instead of showing you the stack bar charts here. I'm showing you a different representation because when you get to 30 factors, which is what we're doing here, it can be hard to see all the different colors. So what we've done here is show a heat map instead of a stack bar chart of 30 different factors. So each column is a factor. Each row, there are lots of rows in this picture, right? You can't really see the individual rows, but each row is a cell. And then the cells are organized by, roughly speaking, cell type, except these cell types are not fact sorted or anything. They're just actually labels that the authors assigned to these cells based on their analyses. And so we're taking that as a useful kind of label, but don't think of it as a gold standard necessarily. Okay, so what you can see is, I know the first factor, as is typical, is loaded on all sorts of different cell types. This is non-negative matrix factorization applied to the transform data. And what you see is each factor, many of them anyway, pick out subsets of cells. So for example, what you see, this second factor is loaded mostly on these cells that the original authors referred to as basal cells from 3 different time points. The data contain data from 3 different time points. If you look closely, you can see some of these cells are much more strongly loaded on this factor than others. Some are almost black, others are almost white here, but it's definitely picking out mostly these cells, and then a few of these proliferating ones. Overall, you can see there's kind of a fairly sparse representation. You can see how that kind of helps interpret the factors. For example, this factor 15 picks out the ciliated cells and just a few of the proliferating ones. So this is a kind of probably a nice, easily interpretable factor. Go and look at the genes involved. Probably you'll see something about which genes are being expressed in these ciliated cells, but although there's another factor that also loads on those cells here. Let me just say that when you get, this is our empirical Bayes non-negative matrix factorization, and it makes things even sparser, and that's because of the priors. And I think that sparsity, well, it changes the results for sure. I think it also arguably makes them easier to interpret because some of the others were a bit messy. There's still some cells that are factors that are not sparse, like this one. And that's okay. Remember that we're estimating a prior for each factor. So it's possible that some of the factors are not sparse, and you estimate a prior that's not sparse, and it doesn't induce sparsity on everything. And while I'm at it, let's just note that this factor 20 here is loaded only on the ionocyte subpopulations. So this factor is kind of picking out that new, quotes, new cell type. There's no factor like that in the original NMF. So that's like, maybe this is, there is a factor here that's loaded on the ionocytes, but it's loaded on other things as well. And this is the topic model. And you can see it's kind of less sparse and definitely different again. So this is non-negative matrix factorization as well, but on the counts. So there are 3 different versions of NMF going on here, kind of NMF on the transformed data, a sparse version of NMF, and NMF on the original counts, not transformed. They produce different answers. So, be aware. This one also doesn't pick out something, the ionocytes specifically. Okay, I'm going to speed up a little bit now and just throw out some more ideas that we've kind of thrown together, put together. I shouldn't say thrown together, it was carefully done. But I'm going to throw the ideas at you now, though. So many of the components here look kind of quasi-binary. Not all of them, but some of them look like they might be kind of on or off, right? They're either present or not. So looking at that, you might think, well, maybe we should have a prior that allows quasi-binary things that are either 0 or non-zero, rather than an exponential, right, having some kind of mode away from 0. Okay. So that maybe we want a prior that encourages that. That's for the memberships. On the other hand, the Fs, that represents So maybe I should say, we're thinking of these K components. We're trying to think of them as kind of representing expression modules or programs that capture what genes are doing together in different cells. So while I think a cell can not have negative membership in a program is either active or not active, the programs themselves could result in genes going down in expression as well as up. So maybe we want to relax the non-negativity assumption on F to allow for that kind of thing. So we're still going to have non-negative on L, because cells are either have the program or don't, or have a little bit of the program, but they don't have negative of the program. And the program can allow genes to go up or down. So now I'm going to let F go be negative or positive. So this is what we might call semi-non-negative, or what might be called and has been called semi-non-negative matrix factorization. And then the other thing we noticed was that if you don't do anything to stop it, the Fs that you estimate may often be quite overlapping or correlated in the genes that are involved. So maybe we'd prefer it if these programs we'd find are a bit more independent of one another. So that might make it easier to interpret if the programs are kind of independent of one another. So we could consider an orthogonality constraint on F. I should say, for a long time, I was kind of against orthogonality constraints, because PCA makes an orthogonality constraint, and it does so as far, basically for mathematical reasons. We thought, well, biology isn't math, and biology isn't orthogonal. But now we're going back to understand that actually carefully used orthogonality constraints can sometimes provide different views of the data, and maybe in some cases more interpretable views. So I'm kind of not orthogonal on both L and F, but on one of the two might be useful. So we've implemented these three ideas into a version of empirical Bayes matrix factorization that, for reasons that will remain unclear to you for now, we call generalized binary covariance decomposition, and that's tentatively accepted in Nature Genetics. So and it's on the bioRxiv, so you can take a look at that if you want to see more details. Just going to show you an example of applying this method to some data. So these data are from pancreatic ductal adenocarcinoma, so pancreatic cancer. And one of the goals of looking at single cell data, these are tumor cells, is to identify subtypes of the cancer. And doing that is complicated by the fact that the data contains strong patient effects. So if you just run a t-SNE or UMAP or some kind of clustering on this data, what you see is that the cells cluster by patient, which kind of obscures any commonality that some of the patients may have, some things in common. Maybe I'll briefly just say we combine data from 3 different studies, so there might be some batch effects here. We'll talk about that later. This is a t-SNE of the data labeled by patient, and the A, B, and C denote the different studies. So what you can, and labeled and colored by patient. So this is what I mean by the fact that, a t-SNE, in a t-SNE, you basically see the cells clustering by patient. And that's the main structure. And that's not wrong. That is an important feature of the data, but it's not all of the features of the data. So that's it. I actually think t-SNE is a useful visualization tool, and UMAP is a useful visualization tool, but I think there's more interesting structure in the data sometimes that doesn't get represented. And we're trying to find that. So this is, again, that kind of heat map representation I showed. So again, the rows are cells organized by the different cohorts, and then within cohorts by patient. So this colored bar here shows the cells that come from the same patient. So this is the first patient in cohort A. This is the second, the green one. The white one's the third patient, etc. And then these are the gene expression programs or components. And what you can see is some of these components, the ones we've highlighted, we've moved to the right here, are capturing mostly cells that are in a single patient. Yeah, so this 34th factor here is capturing the cells that are in this patient. So you can think of these as capturing patient-specific effects. They're not always exactly a single patient, but they're mostly dominated by a single patient. Actually, these patient effects are probably mostly due to copy number variation, and some of that copy number variation is shared across patients, not most of it, but some of it. So that might be why you see some things that are kind of, scattered here. And then these other programs here are ones that don't seem to correlate with a specific patient or a specific data set. Sorry, these two here, this program here, I call it a program. It's a batch effect. It's not a program, right? It's picking out all the cells in cohort C, right? So maybe that's something to highlight here is the method is agnostic about whether a component is technical or biological. It's after the fact you have to go and look at them, try and interpret them and say, well, that one's technical, that one's not interesting to us, but this one is interesting to us. Okay, so the important thing to note here is that in addition to all these patient effects, which is what's dominating the t-SNE, there's lots of other structure here that's capturing things that are shared across patients, stuff that was not evident from the t-SNE. And when you look at the genes involved, many of these factors are dominated by genes that are in processes that make sense from point of view of cancer. And one of the ones that's most interesting is a stress-induced pathway. So one of these programs ends up being dominated by genes related to hypoxia and stress response. And it turns out that pathway, that's this 14 one, is also those genes seem to be associated with survival in a bulk data study. For reasons of time, I'm going to skip over that, and I'm also going to skip over that last picture and wrap up to give you time for questions. So I think matrix factorization has great potential to obtain part-space representations and give you scientific insights, but the way that various assumptions might affect this potential is perhaps underappreciated and also underexplored. And I think we've now developed a set of tools that can help you explore how these different assumptions affect how interpretable the results are. And, I'd love to see them more widely used. I want to acknowledge my co-authors and funding, and I'll be happy to take any questions.
So for questions, if you walk up to the podium and ask on the microphone, that'd be great. Then the Zoom audience will hear. But I will start with a question. Wonderful talk. I really enjoyed the details of the different factorization methods.
I can't hear you very well.
You cannot hear. How about now? Is it better? Okay. Is it good?
Yes, that's better.
Yeah, like from now on, I will always use 30 factors and then say that Matthew Stevens said so. Like when I need to make a choice in the number of factors, it's always going to be 30. But in terms of the sum of parts representation, is there a score that you can use to understand how much your matrix factorization is able to capture that?
Yeah, okay, so the question is, how do you choose the number of factors, and are there scores associated with each factor to understand maybe what they're contributing?
No, more like how well your final factorization is able to capture the sum of parts type of structure rather than just the variance, right?
Rather than just
the variance. Like how well it is, how well it does in terms of finding a sum of parts kind of representation rather than PCA like.
Yeah, can we automatically say, oh, this it looks like a sum of parts, that this is more of a sum of parts than this. And I think
but is there a score like
I think the parts-based representation is phrased in terms of interpretability of the parts, right? So I think it's very hard to automate the process of saying that it's more interpretable than others, because interpretation is kind of in the eye of the beholder. So I don't think I would, I think it'd be hard. In practice, you often have side information, like, on the genes, we know something about genes, right, which the method does not know. And so I guess it's not inconceivable that you might automate the, say, oh, this is more interpretable if it ends up identifying factors where the gene sets are more enriched in those factors, say. You might argue that. But in practice, I'm not sure I'd make a strong argument, but certainly we use that kind of information to try to interpret the factors. Since you asked about K, let me just mention that the empirical Bayes method also has an automated way to select K. So it turns out that sometimes when you fit this model, if the sparsity-inducing families include a point mass at 0 as a special case, what ends up happening is you estimate GL and GF to be point masses at 0, and it basically says, I don't want to add a factor. I'd prefer not to add a factor. Thank you very much. So it does have an automated way as well.
Hi. Thank you very much for the very inspiring talk. I have two questions. The first one, further away. Okay. Start like that. Yeah. So the first question is a simple one. Can you go to slide 10? So generally, in the long lecture metric factorization, do we sort those factors by the importance? And normally the error would be reduced when we have more factors, right? Then why, when go to factor found, the result is worse than using factor 4? Like factor 4 is like 0.013 and factor 5 is 0.015. Was that a problem with the algorithm or?
Yeah, so these are proportional variance explained. Yeah. So in this particular picture, not in all the pictures I showed, but in this particular picture, the factors are displayed in the order in which they were estimated. Yeah. And in general, the signal will decrease. Yeah. But there's not a theorem that says, because we're inducing sparsity here, it could be that you get a very sparse factor, but with a very strong effect in the ones that it occurs in. Okay. So sometimes you'll add a factor, and then the next factor will be slightly bigger in terms of PPE, but maybe it's not as sparse or something. Okay, so it's not guaranteed decreasing. So we don't guarantee that you could reorder them if you want. Instead of ordering them in the order that they were, quote, discovered, which is an algorithmic oriented, you could order them in terms of the proportion variance they explain, and you would then get sparser factors. Okay, so this is on order one. Thanks. The second question is regarding the whole problem for the non-matrix factorization. I think contains like a right term is x times y, which is bilinear. So that means the whole problem is non-convex, which means the solution may be non-unical. And if you go to slide 21, then in that case is how, I mean, the result maybe depends on the initialization or the constraint you put like a sparsity. And when we apply to this very important generic application, is there any way or can we guarantee to get like some stable, unical, good solution for those bases or factors? I mean, if we cannot do it in NMF, can we do it in more better in Bayesian factorization? Because it's really important real application. We want to get a stable and unical good solution. Great question. So you're right. These are non-convex optimization problems. Even vanilla NMF is non-convex and so can depend on the initialization. And so the way people typically deal with that is to run it multiple times and then either to choose the one that has the highest log likelihood or to do some kind of consensus thing. There are various things people do to try to deal with that. And the Bayesian, whether the Bayesian approach is more stable or less stable, I guess it's possible it's slightly more stable than the priors, but it's still a non-convex optimization problem, and it's still going to depend on initialization. We have a specific initialization we use that seems to work well often in practice, but could maybe still be further improved. Let me just mention, apart from PCA, almost every other approach to dimension reduction used in practice has this problem. So clustering has this problem. Almost any clustering algorithm you're going to have is going to have this problem. T-SNE has this problem. Even UMAP has this problem. They're all like, they're non-convex optimization problems. So all I can say is that we found initialization approaches that produce good, useful results in practice on a consistently, although, as I say, there could be room for further improvement.
And that's for the Bayesian approach or the traditional matrix factorization approach?
That's for the Bayesian approach. So I'm claiming that. I think that this NMF was probably run from a random initialization. You're right. We didn't try hard on choosing a good initialization or even doing a consensus approach.
So do you think it's more probability or it's more easy, it could be handled by Bayesian approach, by including those priors and those?
Move on to the coffee break.
Sorry.
To answer the question, I think maybe, but I'm not sure.
Thanks.
Yeah. So we have
Your question, I'll just follow it if I could.
yeah. So
Thank you
we have like
for
several
joining,
people who want to talk, but my no, you go ahead and then something.
Thanks for coming. Thanks for presenting an updated view on Empirical Bayes. Can you just comment very briefly on how you handle, what's your preferred way of handling the tau?
Yeah, okay, the precision parameters. Yeah, my preferred way, so that's the kind of thing
This one, yeah. Those guys.
yeah. My preferred way is to have a gene-specific tau for single-cell data. The software allows row-specific, column-specific. It also allows other more fancier things, but they tend to slow things down quite a lot. So in practice in single-cell data, I use a column-specific tau, and then you also need to do a little bit of regularization of the taus, because otherwise there's a danger that some column will get a 0 tau, and basically it will try and fit. So we add a little bit of regularization or fudge factor to stop the taus going to 0. Infinity, I guess. Infinity, I meant, yeah.
Great talk, thank you. Do you have any, for your single cell experiments, do you have any specific pre-processing steps? Like, for example, you can try to remove genes which could be met in all the cell types or something like that.
Right, great. So the question's whether we do any specific pre-processing, and the answer is no. Essentially, we do very limited filters. Possibly we remove things that are expressed in a very small number of cells, mostly just for so to reduce computation, because we think they won't add very much information. But we do minimal, let's say minimal pre-processing typically. I think we think that genes that are expressed across all cells, that will come out potentially in one of the early factors. And we don't, some of those genes, while being ubiquitously expressed, may also show differential expression across different cell types. So we don't really want to remove them a priori. Our belief is that if they don't show changes in expression, they'll just appear in one factor and then never appear in any others. So we think the method should deal with that automatically, is the bottom line.
Okay, thank you.
Is there maybe one more question?
So you should have to decompose matrix factorization for the EQTL results with GTEx cells. Now with single cell EQTLs, in principle, we can also do that, right? But however, for single cell, you have different cell types, and many comes with a lot of cells, and some could come with very few cells. So how do you account for the imbalance of the number of cells, and therefore for example, the difference in the uncertainty of the estimation of the effect sizes?
That's a great question. So yes, in in the model here, the x's may actually be measured with some error and may be different for different observations of x. So you can actually modify this model to have an additional error term to capture that, right? So the model is x equals LF transpose plus E plus a measurement error term. And that measurement error term has at least, so what distinguishes it from E is we're going to assume that that measurement error is well characterized. That is, we already know what the variance is. So if you change this model to add another error term that has known variance and which may be different for each observation, the software will do that. It does make the computation quite a bit heavier, right? So that's, yeah. So we can deal with that. It's a great question, but it's a lot slower than if you, yeah. So there are sometimes we just ignore it for reasons that we want to get an answer, you know, today and not in a week.